everyone, welcome to today's class. Today we are going to study about RNA transcription, that means the synthesis of RNA from DNA under the subject biochemistry BP203T. Let us first of all understand the concept of transcription. In transcription we have seen DNA is replicated and then DNA to RNA, the process is called as transcription, and later on in the next lectures we will study about RNA to protein. This is the conventional concept. What is the current concept? That means when we are studying about DNA, we are studying about genomes. That means genomes replicates themselves. Then through genome we are forming transcriptome. This process is called as transcription. Then later on transcription will form proteome. This process will called as translation. This is the current concept of RNA transcription. Now let us for that matter we need to understand what is genome, what is genomics, what is transcriptome, proteome and so on. So genome is the total DNA combined in organism or the cell. It includes the chromosomes in the nucleus, DNA in the mitochondria and the chloroplast in case of plants. So the whole thing, the whole DNA having in an organism is called as genome. The study of these DNA and the rest of the things like chromosomes, chloroplast, mitochondria, everything, when we study about it, the subject will be called as genomics. Then the transcriptome, the RNA copies of active protein that coding genes, it is initial product of gene expression, it is called as transcriptome, and the study of this transcriptome will be called as transcriptomics. When we study about something, we call the subject with that name only. That means when we studied about genome, we call it genomics. When we studied about transcriptome, we called it transcriptomics. Now the proteome, as the name suggests, it has something called as protein in it because the word proteome forms from the word protein. The cell storehouse of protein with their nature and biological functions. What is proteomics? Definitely the study of proteome is called as proteomics, and metabolomics is one more important thing, the use of genome sequence analysis for determining the capability of cell tissues or any other organisms to synthesize a metabolite is called as metabolomics. Next, now let us discuss about the transcription. Transcription is the process of copying the genetic information from one strand of DNA into RNA. So when we are synthesizing RNA from DNA, we call the process transcription. One DNA strand serve as a template and produce the working copies of RNA. That means the full DNA, like we do have the we do have the DNA, two strands of DNA here. Only one DNA strand will work as a template for the synthesis of working copies of RNA, and then it is called as the template strand. The other strand which will not participate will be called as coding strand. So the strand which is participating is called as template strand, and the strand which do not helps in do not participates in the process of transcription, it will be called as coding strand. Okay. So as we can see in the structure given here that there is one template strand and one coding strand. Next, the transcription is very selective. RNA are synthesized for only some selected regions of DNA. That means it is also a very selective process as DNA replication. Entire DNA is not expressed in transcription, only the certain parts of DNA, only the certain regions of DNA is exposed for transcription process. For certain other regions DNA there must be no transcription at all. The product formed in transcription is called as primary transcript. Later on, after doing certain modifications, certain changes, they become functional, and those changes are called as post-transcriptional modifications. Changes like splicing, terminal additions, base modifications are done once the primary transcripts are ready. That means we have done the step of transcription, we have our product here, we call it primary transcript, but after the formation of primary transcript we need to do some modification with the primary transcript to make it functional RNA molecules, then the step will be called as post-transcriptional modification. Then let us start with the transcription in prokaryotes. So, in prokaryotes, a DNA dependent RNA polymerase is the single enzyme for catalyzes the transcription process for all type of bacterial RNAs. Like RNA polymerase of E. coli, it is a complex holoenzyme with 5 polypeptide subunit. Which are the 5 polypeptide subunit? 1 alpha, 1 beta, 1 beta dash and 1 sigma factor. That means all these factors are important for the synthesis, for the transcription to happen. Okay. So, now the what are the steps involved in here? The steps are initiation, elongation and termination. So, the process needs to be initiated, started, then elongated, that means there will be the elongation of the RNA molecule, the primary transcript, and termination means when we get the desired length of that RNA molecule, the process needs to be terminated then and there only. So, the whole thing will be divided in the 3 steps: initiation, elongation and termination. As the DNA, as we can see from DNA molecule, there is a synthesis of RNA, RNA molecule is happening. So, here you can see this is the first strand of DNA, this is the second strand of DNA. With only one strand of DNA, with the help of only one strand of DNA, there is a formation of this RNA molecule happening. The rest of the other DNA strand is not participating, that means the coding DNA strand is not participating, whereas only the template strand of DNA is participating. So, this is the template strand of DNA which is participating in the synthesis of newly RNA strand. So, here you can see there is a synthesis of RNA. That means it is happening in here. Later on we will discuss about the elongation and termination process. So, initiation process, RNA polymerase binding to the promoter site of the DNA. So, DNA has one promoter site, likewise suppose this is the promoter site in here. So, the RNA primer RNA polymerase binds to the promoter site of DNA. There are 2 base sequences of coding DNA strand, that is sigma strand, sigma factor of the RNA polymerase, which can recognize it for the initiation of complexes. Like I told you there are 2 sequences which control this thing. First is the Pribnow box, also called as Tata box, and the second is your 3-5- sequence. The sequence is very important. Every time when we are doing DNA replication, transcription, translation, we need to look after the sequence also, because it is very important to see from where we are starting with the process and where it is ended. So, there are 2 base sequences on coding DNA which helps us to initiate the transcription process. First is the Pribnow box, also called as the Tata box. In this, it consists of the 6 nucleotides which is in sequence of TATAAT, and it is located on the left side about 10 bases away from the starting point of transcription. So, we know the space where it is located and we know the sequence which is TATAAT and helps in the initiation process. And the second is the 3-5' sequence. Region on promoter site of DNA, it will be there. Second, it contains a base sequence of TTGACA, which is located about 35 bases away on the left from the transcription side. That means these two are very important for the process of transcription, for the initiation of transcription actually. Now, the elongation. In elongation, the RNA polymerase recognizes the promoter site. That means the sigma factor is released and transcription proceeds. RNA synthesized from 5' to 3' sequence. RNA polymerase utilizes the ribonucleotide triphosphate. Which triphosphates we are here? We are studying here ATP, GTP, CTP, UTP, and likewise for the formation of RNA. Sequence of base in mRNA is complementary to template of DNA. That is because we are here, we are studying RNA here. So, here will be A, U and G, C. So, A, T will not be there, A, U will be there. RNA polymerase is different from DNA polymerase. How? Because in RNA polymerase we do not require, in transcription we do not require primers, we do not need endo and exonucleasing activity. RNA synthesis does not require any proofreading, because if there will be any mistake happen, that is not that much lethal, that can be rectified later on also. And topoisomerase overcome the problem of supercoiling here. So, in the termination process, there are two types we can terminate the process, like the first is row dependent and the other one is row independent. The row dependent means there is a factor, that is a row protein is there which binds to the growing RNA. When we got the desired length of RNA we want, then this row factor will bind that growing RNA. It acts as ATPase in the bound state and finally terminate the steps there and there only. So, there will be the release of RNA. The second step, how the termination will proceed? The row independent step, the row independent termination. In this, there is a formation of hairpin like structure. Hairpin like structure means there are the sequence of palindromes. Palindromes are those words which have same starting and end point. Okay. So, these are, when you read from the starting, it will go to nowhere else. It will go to the end point, again you study, you read it from the end point, it will go to the first point. So, there you are not growing to a RNA synthesis. So, here we can use these two techniques for the termination. We are studying these two techniques for the termination. First is your row dependent and second is your row independent. This is the diagram which is telling us the full process in which the initiation, the elongation and the termination happens. As we can see, this phase is our initiation phase. In this phase, the sigma factor attaches with the RNA and it has start synthesizing in this whole process. In here, when there is a starting, there is the initiation of RNA strand happens, then there should be the elongation. So, our respective enzyme helps in the elongation. When like this, you can see here that the RNA strand is elongating, like here it is again elongated. Now, we have the desired length of RNA strand. Now, we need to terminate it. So, how we are terminating it? There are two steps we just studied about, row dependent and row independent. When there is a row factor happens, it comes and attaches with the newly synthesized RNA and there termination will be identified and terminate, the growing chain of RNA will be terminated. So, we have got the full process here. Now, let us discuss about the transcription in eukaryotes. The process or transcription in eukaryote is a little complicated than prokaryote, because we all know prokaryote are mostly very easy single organelle compounds, whereas eukaryote has each and every organelle in them for specific purposes, for specific functions. So, eukaryotes are having tough structures, tough life cycles than a complex life cycle than prokaryotes. So, there are certain different enzymes are here for doing the process. So, in there are certain enzymes like RNA polymerase. In RNA polymerase, there are 3 types of RNA polymerase. RNA polymerase 1, which is responsible for synthesis of precursors for large ribosomal RNA. RNA polymerase 2, in this synthesize the precursor for mRNA and small nuclear RNA. RNA polymerase 3 for the formation of tRNA and small ribosomal RNA, and mitochondrial RNA polymerase, that is resembles prokaryotic RNA polymerase in structure and function. So, we have seen there are 3 types of RNA polymerase and 1 mitochondrial RNA polymerase is also there. All the polymerase, RNA polymerase are functioning in different manner. Next is there are the promoter site. There are 2 types of promoter site here. One is the Hogness box and the again is CAT box, that is CAAT. So, Hogness box is similar to Pribnow box in the prokaryotes. We have studied now, and it is located about 25 bases away from the starting of mRNA synthesis. And CAT box is, it is located between 70 to 80 nucleotides from the starting of transcription, and these 2 sites help RNA polymerase to recognize the requisite sequence on DNA from transcription. So, these 2 boxes, these 2 sites are called as promoter site, which promotes the RNA synthesis or transcription in your eukaryotes. These are the transcription factors which are very important for the transcription process. So, a large number of transcription factors are needed with the promoter region of eukaryotes. In human, there are 6 different types of transcription factors, namely TF, that is transcription factor 2D, transcription factor 2B, transcription factor 2A, transcription factor 2F, transcription factor 2E and transcription factor 2H, as the diagram also suggests the same. So, here at this position the transcription factor helps in promotion in the synthesis of RNA in the transcription of RNA. So, we have here the promoter sites, the activators which activate these things, and the enhancer. Everything will be working in cooperation with each and every one. So, the enhancers are those which increase the gene expression about 100 folds. Now, next is heterogeneous nuclear RNA. Heterogeneous nuclear RNA are the primary mRNA transcript that are produced by RNA polymerase II in eukaryotes, is often regarded as hnRNA. That means heterogeneous nuclear RNA is also called as hnRNA. This is then processed to produce mRNA needed for protein synthesis. Once we have done with all the things, then there will be, once we got our RNA primary transcripts, once we got, then we need to do the post transcriptional modifications. Now, what are post transcriptional modification? Post transcriptional modifications, as we have discussed, that after the synthesis of primary transcript, there should be some kind of modifications done with the primary transcript to make it a working RNA, primarily working RNA. So, for that we are doing like terminal base additions, base modifications, splicing, these are called as post-transcriptional modifications. Primary transcript converted into active forms after this process. Ribonucleases are responsible for processing rRNA and tRNA in both prokaryotes and eukaryotes. So, likewise, after the synthesis of the primary transcript, after the synthesis of RNA, there needs to be some sort of changes, there needs to be some sort of modification in the primary structure to get the functional, to get the active RNAs. Okay. Next, the prokaryotic mRNA synthesized in transcription is almost similar to functional RNA. Whereas in eukaryote, what was happening, that when we form the primary transcript, we definitely need to go for post transcriptional modification to get our active forms of RNA. But here when we are synthesizing, it is almost similar to getting functional mRNAs in the eukaryotes. The 5' capping, the 5' end of mRNA is capped with 7-methylguanosine by an unusual 5' 5-triphosphate linkage, whereas 5-adenosyl-methionine is the donor of methyl group. So, here we can see from where we are getting the groups, that means we are discussing here the post transcriptional modification. Post transcription we are studying here, like how and what groups we are incorporating here. So, 5' capping means when we are adding, when the 5' end of mRNA is capped with 7-methylguanosine by the unusual 5' 5-triphosphate linkage and 5-adenosyl-methionine is the donor of methyl group. The cap is required for translation besides stabilizing the structure of mRNA. Now, second modification will be poly-A tail. A large number of eukaryotic mRNAs possesses an adenine nucleotide chain at the 3' end. The poly-A tail as such is not produced during transcription. It is later added to the stabilized mRNA. However, poly-A chain get reduced as the mRNA enters in the cytosol. So, when we are incorporating this poly-A tail, there is again, that means at the initiation process of this modification and the later on there are some changes happening. And when we are there is a removal, there is a process called as intron and their removals. What are introns? First of all, let us understand what are introns. Introns are the intervening nucleotide sequences in mRNA which do not code for proteins. So, on the other hand, exons of mRNA possesses genetic code and are responsible for protein synthesis. When we remove these introns, remove this by using small nuclear ribonucleoproteins particles that are also called as SNRNPs. These are the, again I am telling you what is the full name, small nuclear ribonucleoprotein particles, SNRNPs. They are in terms are formed by the association of small nuclear RNA, snRNA with proteins. So, the introns are those substances, are the intervening nucleotide sequences of mRNA which definitely do not code for proteins, and exons are those which possesses genetic codes and responsible for protein synthesis. So, introns should be removed when we are doing the modifications because there are no use of it. The term spliciosome, this is again our modification, our post transcriptional modification. The spliciosome is used to represent the SNRNA RNP, SNRP is also called as, is pronounced as SNRP, association with hnRNA at the exon-intron junction. Post-transcriptional modification of mRNA occur in the nucleus. The mature RNA then enters the cytosol to perform its function, translation. As you can see the full structure here, the full transcriptional post-transcriptional modification is there. So, the end modifications, when we are doing the end modification, there are two type of end modification, that is 5' capping we studied earlier and poly A tail changes. Then the splicing, in this we are doing introns removal, there is cutting, it is as simple as to understand. Then there is a wrong implementation of a nucleotide, they need to be cut that part and that is again resealed. So, we need to cut the wrongly incorporated part, because RNA, when there is an incorporation of a wrong nucleotide, it is not that dangerous as in case of DNA replication. So, it is done, it can be done post in the post transcriptional modifications also. And there, if there is any chemical modification, that means if any new chemical groups are added or removed. So, there are these type of changes we can study. Now, the inhibitors of transcription. So, there are certain medicines which are used for the inhibition of transcription. Why we need to inhibit these processes? Because bacterial DNAs, bacterial RNAs somewhat are dangerous if they are growing inside our body. They can cause certain diseases. So, to stop that we need some inhibitors. So, actinomycin D, also called as dactinomycin, this is the first antibiotic used for the treatment of tumors. It blocks the movement of RNA polymerase, hence it inhibits the transcription process. Second is rifampin, that is the treatment of tuberculosis and leprosy. It inhibits the activity of RNA polymerase beta subunit in prokaryotes. And the third is alpha amanitin. This is helps in to bind with RNA polymerase 2 in eukaryotes and inhibit the transcription. These are the references which I have used. Hope you have enjoyed today's class. Thank you so much.